Hello, everyone. I would like to welcome you all to the CCG webinar series. Thank you for joining us. Today's speaker is Valerie Campagna-Slater. Valerie is working as a senior application scientist at CCG in Montreal. My name is Barbara Sander. I'm also a senior application scientist. I'm working for the CCG office in Germany, and I will be moderating today's webinar. If you have any questions during the webinar, please use the chat window to type in these questions. You can open the chat window by clicking on the speech bubble button, which is located at the bottom right of your screen. Make sure to send your questions to the host. That will be me. We will try to answer the questions at the end of the session. So without any further delay, I'm very happy to introduce you to Val, who will present approaches and applications of in silico fragment-based drug design in Mo. Val.
Thanks for the introduction, Barbara. So as my colleague just mentioned, today we'll be talking about in silico fragment-based drug discovery. So we'll start with a brief introduction on in silico FBDD methodology and fragment databases. And then we'll jump into a live demo where we'll look at active site analysis and then different types of fragment-based drug discovery approaches. So we'll be looking at fragment growing, linking, scaffold replacement, and medchem transformations. Throughout, we're also going to look at how we can use Pharmacophore models to design compounds that would have specific interactions within the binding site. So that being said, what is fragment-based drug discovery? So the idea here is to identify fragments of low molecular weight that can weakly bind to a specific target, and then optimize these fragments into a drug candidate through a multi-step process, which is depicted at the middle on the middle of my slide here. So the idea is we'll start with a protein or receptor structure and a database of different, or a library of fragments, which we're going to screen to identify some initial hits. From this, we're going to do some hit characterization. And ultimately, what we would like to do is get some X-ray crystal structure of these fragments, either one of them or multiple fragments bound to this receptor. And then we'll be going on to fragment optimization. And this is going to be the main topic of the talk today. So we're going to be looking at different approaches in silico, of course, that we can do some fragment optimization. For example, linking, growing, and replacements. Once we have some lead candidates like this one, we can then, of course, do some lead optimization to design some drug candidates. So in silico FBDD can attempt to mimic several of these steps. And as I said, we're going to be focusing mainly on this one right here. Now, some of the advantages of FBD in silico or FBDD in general include better coverage of the chemical diversity space. So these fragment libraries can contain very diverse building blocks, if you will. Higher compound hit rates compared to larger conventional ligands. And so we're not looking necessarily to fill the entire pocket, but just sub-regions of the, or very localized regions of the receptor. And finally, we can elaborate from these initial fragments to achieve high binding efficiency. So typical in silico FBDD methodology might look something like this. So as I mentioned, we'll start with a crystal structure with some fragments bound to it. And from this, we're going to generate some initial queries. We're going to search a database of different 3D fragments to construct some molecules. Now, of course, we can generate a whole bunch of molecules. What we can do is to filter them. So for example, for the molecular properties, we want something, for example, that's lead-like. If we have any QSAR models, fingerprint models, or Pharmacophore queries, we can use those as well to filter down the molecules that are being constructed. Next, because we're doing this in the context of a receptor structure, we can do some refinement. In other words, we can do an energy minimization directly in the binding site. And from this, we can do some scoring. So we can score, for example, by predicted binding affinity or by synthetic feasibility. And that will allow us to rank order the different hits that we're going to get. And then, of course, we finish off by visual inspection of the highest scoring or the most promising compounds to decide which ones we want to move forward with. So what do we need to get started with in silico FBDD? Well, basically, we need three things, three components. First of all, we need a receptor with some fragments bound to it. It can be one fragment or two fragments like this or more. Ideally, we want a co-crystal structure. That's the ideal starting point for optimizing a ligand or fragments into a ligand. That being said, if we don't have a co-crystal structure, if we're starting with an apo structure, there are approaches to position some fragments in the binding site For example, we can think of docking. Now, that's beyond the scope of what we're going to cover today, but do note that that is possible. Now, today we're going to be looking at the case scenario where we do have a co-crystal structure with some fragment bound to the receptor. So that's the first component, a reliable starting point. The second one is a database or library of different fragments. So you can think of fragments, linkers, scaffold, these different building blocks that we're going to use to then reconstruct new molecules. And finally, the third component, we need a computational tool, so a method to actually combine the initial fragments with these fragments from the linker or fragment database into new molecules. And so that's going to be the main topic, of course, today. Now, before I elaborate on this, I just want to come back to the topic of fragment databases really quickly. So how do we construct these databases? Well, the idea here is typically we'll be starting with some input molecules, which are organic or lead-like molecules. You can think, for example, of taking the zinc library, enamine, or chemical libraries as a starting point. From this, we're going to generate different tautomers and protomers for each of the molecules. And then we're going to run a fragmentation protocol. So basically, taking these molecules and breaking them up in smaller subblocks. And of course, there's a number of rules to do that. Once we've done this, we can filter that list of initial fragments because some of them might not have very good properties. Some of them might have reactive groups and things like that. So we can filter those out. Once we have that, we, of course, remove duplicates because you can imagine as we're starting with a bunch of different lead-like molecules, some fragments are going to occur more than once. Once we have a unique fragment list that has been filtered out, we then run a conformational search on each of these fragments. So ultimately, our fragment database that we're going to be using is not just containing a bunch of fragments, it actually has an ensemble of conformations for each of these fragments. So you can imagine that we're starting with a fairly large number of molecules. We're going to have an even larger collection of fragments and potentially very, very large number of different conformations for each of these fragments. So there's different types of FBDD methodologies here. I've listed some of the ones we have in Mo and which we'll be covering today. So first of all, we're going to be looking at fragment growing. So the idea here is, let's say we have a receptor, we have a fragment bound to one specific subregion of our pocket. That's the yellow circle right here. How do we grow out of this region to fill adjacent subpockets or areas in our receptor? Next, we'll be looking at fragment linking. The idea here is if we have multiple, 2 or more fragments bound to a receptor, how do we actually link them, connect them together to generate just one single molecule? Next, we'll be looking at scaffold replacement, where we have one molecule and we want to replace a central core with something else, for example, to optimize the affinity or its properties. But of course here, the key component is to keep the geometry intact for these R groups. Finally, we're going to be looking at medchem transformations. The idea here is to tweak the properties or the affinity of a given compound with very minor localized modifications. And often these are based on the concept of bioisosteres. Now, finally, a last option, which we won't be covering today, is R group screening, of course. So generating combinatorial libraries of compounds based on multiple R group substituents. So that being said, let me minimize my slides and we'll actually jump into a live demo. So in this example, I'll be loading in a structure of P38 MAP kinase. So I'll click on open here, select my structure, and typically you'd be opening up a crystal structure, which means the very first step is always to prepare the structure. What do I mean by that? Well, typically a crystal structure won't have any charges, probably no hydrogens. There might even be some missing side chains or some missing loops. And so in the right-hand side button bar, we have the quick prep tool, which is available to do that. Now, in this case, for the purpose of this demo, we've already prepared the structure. So as you'll see here, there's already hydrogens and charges on this molecule. So I'll just go ahead and hide the ribbons, and we'll start by analyzing the pocket. So before we can do any type of FBDD design on this initial fragment, we do need to know what's the pocket like, where can we modify this ligand. Now, the ligand or the fragment here is shown in cyan and in stick mode, and in wire and gray carbons here, that's our receptor. And as I rotate, you can kind of imagine that this fragment here is fitting in a bigger pocket. But it's hard to imagine just like this. So the very first thing I usually do once I've prepared my structure is to generate a surface around the receptor to get an idea of the state. So where can I actually build something off of this fragment? Where is it binding tightly, and so on and so forth. So to generate some surfaces, we can go back to the right-hand side bar where it says surface. We have access to some quick surfaces. And at the bottom here, if I click on surfaces and maps, this opens up the full panel. So from here, I can choose different types of surfaces I want to create. In this case, I'll just select the default, which is molecular surface. I can customize where I want to generate the surface. So I'll be generating the surface around the receptor atom, but I'm going to extend a little bit further away from the ligand. So I'll increase the radius to 6 angstrom. The bottom here is for rendering options. For now, I'll leave it as is, and we can modify that afterwards. So as I click apply, we have the surface now that appeared. You'll notice it's mostly gray, and that's simply because the carbon atoms of my receptor are colored gray, and so the receptor is now mostly colored gray. That being said, we have some blue here, some dark blue, and that's where we have some nitrogen atoms on the side of the receptor, for example, this arginine. Where I see red, that corresponds to the position of oxygens. And of course, at the bottom here, this yellow patch, that would be my sulfur from the methionine right here. You'll also notice these white patches and these cyan patches. These are indicating the location of polar hydrogens and lone pairs of electrons. And so that's for when I'm trying to optimize a ligand, I could potentially target this. That would be the polar hydrogen on this lysine, or these lone pairs here for new interactions. Now, in this case, we won't be using that information. So I'm just going to go ahead and change the color and set that to a constant white color. Okay, so let's have a look at how this fragment is binding. So as I rotate, we can see the fit seems to be pretty good at the bottom here. It's filling out this bottom pocket really well. If I rotate upwards, we can see that there's actually more space on the top here. So as I'm analyzing my system, thinking of ways to optimize this fragment, one thing that might come up is the idea of extending this fragment in this area. So this is something I might want to fill. Now, if I rotate the structure, we'll notice there's actually a second pocket on this side right here. And turns out if I rotate even more, we can see that the two pockets are actually connected via this sort of tunnel right here. So this is another opportunity. Perhaps I could add something at this position and try and extend out in this region right here. So if I rotate again, that would be this hydrogen right here. Now, another convenient way to see where I could potentially grow a ligand is to use the concept of R vectors. So if I go to the ligand menu in the right-hand side button bar and I toggle on ligand R vectors, this will display some light green arrows at the position where there's enough room to add at least something the size of a methyl group. And so again, if I rotate down here, we can see that this is a really tight fit. There's no R vectors here. And in fact, there's 3 positions where I could add something. Those would be the 2 hydrogens on the phenyl ring here, as well as this one right here, which again is that position we had identified, which can link into this or grow into this region right here. So that's exactly what we're going to do. We're going to try and grow this ligand out into this region. Now, before I do that, let me just clear up the view a little bit. First of all, we can't really see because of the surface here. So I'm just going to make the front of my surface transparent, just like that. That way, as we try to grow into this region, we can see what's going on. So that's the first thing I'm going to do. So I'll minimize this for now. I'll hide my R vectors as well. And lastly, maybe as a starting point, I want to know what are the properties of my starting structure. And so I'm just going to go toggle on the ligand properties. As I do that, in the top left corner, you're going to see some properties listed: molecular weight, TPSA, log P, log D, log S. And so now as I'm designing new compounds, I can keep track of these properties and see if I'm going in the right direction. Okay, so that being said, let's actually start growing this ligand with the fragment-based drug design tools. So we'll go to the compute menu and I'll click on fragments. And as you click on fragments, you get a full list of all of the FBDD tools we have in Mo. Now, it turns out most of these, at least the top 6 or 7 here, will all open the same panel. Now, in this case, I want to grow this, this initial fragment, so I'm going to click on Add Group to Ligand. This opens up the unified fragment-based tools, which basically is separated in 4 sections. The top 2 sections here, those allow us to define those 3 main components I talked about in my slides, right? So we have to have a receptor structure with some initial bound fragments. That's the top section here. We need a tool or an operation that we want to do. In this case, that's going to be add group to ligand, but I could switch back and forth between the different FBDD tools. And we need a linker database. I'll come back to that in a second. The third section right here That's for filtering. So different ways to filter based on descriptor filters, so molecular properties, model files. For example, we can think of QSAR models or fingerprint models, as well as Pharmacophore queries. We'll come back to that later. And then at the bottom here, we have a section for minimization or refinement options, scoring, as well as output options. So let's move this to the side, and we're going to set up our first set of calculations. So going from the top, by default, it sets the ligand to be my current ligand. That's fine. And the receptor atoms, if I want to double check, I can click on the question mark. Those will be my receptor atoms. In other words, I'm going to do the fragment growing within the context of the receptor. The operation, I could switch back and forth, but of course, I've already selected add group to ligand. That's OK. And now I can define which linker database I want to use. So a database of different fragments that are going to be used to try and attach at this position and grow into this area. Now, we do provide a default database, this one right here with Mo. So when you start using the tool, you can use this default database. We also have a larger database. This one is mostly for testing purposes. We have much larger databases you can use as well. The third option, of course, is to provide your own fragment or linker database if you have one. And so in this case, that's what we're going to do. I'm going to click on the folder icon, and I'm going to go select this linker database we've prepared for the purpose of the course today. So I'm going to double click on it. As I do that, you see it's added at the bottom. So right now, if I clicked OK, I'd be actually going through or screening two different linker databases. Now, in this case, I'll actually remove the default one. We're just going to use the one that we've prepared for today's webinar. And just to give you an idea, it contains about 40,000 different linkers, each of which, of course, has an ensemble of different conformations. So we'll go ahead and click OK. As I do that, you'll see the linker database has been updated right here. Next, we can add or modify some filters. For now, we'll just keep the default as is. And at the bottom, the only thing I'll change for now is the output database name, just so we remember which experiment this was. I'll call this grow.mdb, since I'll be growing this fragment into the neighboring pocket. So now the last thing I need to do is actually define my query. So I just have to specify where I want to add a group. And that's going to be this position right here, which is pointing in the right direction for this tunnel right here. Now you'll notice as I selected it, we have a bright green arrow that appeared. This is telling me what's the attachment point or the exit vector for my query. And in the panel, it tells me one connector, one essential. And so I'm ready to go. I can go ahead and click search to launch the calculation. As I do that, you'll notice the database viewer appears. And so this is where my output structures are going to be collected. The database viewer looks a little bit like an Excel spreadsheet. The main difference is in this mole field, you'll actually see the structures of our compounds. And the database viewer, of course, is also the main launch point for everything related to cheminformatics and more in Mo. Okay, so just one more word about descriptor filters. So right now we've used the defaults here, which means that we're only going to be keeping molecules that are generated which have a molecular weight below 500, a TPSA between 40 and 140, and no reactive group. But of course, you can customize these, so you can add some more descriptors, any of the descriptors that are available in Mo. You can also specify some smarts matches. So if there's a specific group you want or you don't want in your compounds, you can also add those in here. Now, any descriptors that are calculated here, you'll notice in the output database, they're also going to be listed here. Okay, so that being said, you can see some hits are starting to appear, and we do have a progress bar at the bottom here telling us how many linkers have been screened, how many structures have been generated, and if I expand these, we can see the molecules in 2D. Now, of course, what's more useful is to actually see it in 3D within the context of the receptor, and so for that, we can go to File, Browse to open the database browser. So I'll just move this to the side here, and I'll change the rendering so we can so it pops out a little bit more. I'll put this in ball and stick and gold color. And so this is the first compound that we designed using the fragment-based tools. So we can see that the fragment here is exactly on top of the initial fragments, but then we have new linkers that are or new R groups that are being attached. And so I can use the browser now to browse through the different structures which are suggested. And as I browse through, I might find some, for example, this one, that are making novel interactions with the receptor. So for example, here it's making a hydrogen bond with this residue right here. If I select it, I can see that's glutamate 71. So these are interesting compounds, and I can scroll some more and see if I get some other interesting ideas. Now here, a lot of the compounds I'm generating are not making any specific interactions with the receptor. And so one of the things we can do when we're trying to grow a ligand is to target some specific interactions, some interactions with specific residues in our receptor site. So the way we can do that is with a Pharmacophore query. So that's the next example we're going to look at right now. So I'll go ahead and close the browser, and I'll close the database as well and stop this search. So now what we're going to do is we're going to redo exactly the same query, but we're going to add a Pharmacophore filter to make sure that we're going to create some new interesting interactions with a receptor. Now, if I already have a Pharmacophore query, I can go click on Browse and open that up. In this case, I'm going to create one on the fly, so I'm going to click on Editor here, which opens up our Pharmacophore Editor panel. Now, let me just move this to the side so we can see our ligand again. And as I do that, you'll notice that when I open the Pharmacophore Editor panel, there's these annotation points that's also appeared on our structure. These dots are abstractions of the types of chemical features we have on our fragment. Now, I'm not so interested in the annotation points on this fragment because I'm not going to be changing this fragment. What I'll be doing is growing into this region right here. So my Pharmacophore query won't be on the fragment itself. It needs to be somewhere in this region. So what I'll do is I'll hide the ligand annotation points by clicking on the yellow L button right here. And instead, I'll display the receptor annotation points by clicking on R right here. So R stands for receptor, S for solvent, and L for ligand. So now what I'm seeing in the Mo window are some annotation points based on the receptor. So not on the fragment, but on the receptor. So this is telling me what the receptor would like to see as a binding partner. And so again, we're trying to grow out in this region, so we'll just focus on this spot right here. So I see three annotation points. First of all, I see a donor annotation point here. This is telling me that this is the optimal position to place a hydrogen bond donor as I'm building a new molecule in this area so that it can hydrogen bond to this glutamate right here. So just like in our ligand that we spotted a few minutes ago. So what I'll do is I'll click on feature. I've selected this dot. I'll click on feature to create a feature at this point. Similarly here, I see an acceptor point. So I'll click on feature to create one here. So this would be now the optimal position to place an acceptor if I wanted to make a hydrogen bond with this backbone nitrogen right here, the receptor. Now I'll keep it simple. We'll just do a 2-point Pharmacophore for now, but I'll just point out that there's also a hydrophobic annotation point right here. So that would be another option, right? So there's some need to have some hydrophobic group in this region right here. For now, I won't create a feature, but we'll keep that in mind for later on as we're designing different compounds. Now, by default, the radius of these spheres is 1 angstrom, which means it's a pretty tight fit. So the donor and the acceptor really need to fit exactly in these positions. If I want to be a little bit more flexible, I can also increase like this the radius of these these features. So I'm going to be a little bit more flexible in terms of exactly where that donor and the acceptor need to be. Okay, so that's my Pharmacophore query. That's what I'm going to use. And so now when I run exactly the same query, I'm only going to keep compounds that have a donor here and an acceptor here. Now, typically I would save this query so I can reuse it later. But for now, just for the purpose of speed here, I'm just going to minimize this, and you'll see that where it says Pharmacophore here, it's already linked to that Pharmacophore editor. So it will be using that query. And perhaps I'll just hide the annotation points to clear up the view as well. Okay, so we're going to be rerunning exactly the same query. So I'm going to select the attachment point. Again, the arrow's green, indicating everything's okay. And so I'll just click search to rerun the calculation. It asks me if I want to overwrite. In this case, yes, I'll just overwrite our previous database. So the output's going to look the same, except now all the compounds will have a donor and an acceptor in the right positions. And if I scroll to the side here, we also have a new column called Pharmacophore RMSD, which is telling me how well the compounds are matching up with this Pharmacophore query. So let's have a quick look. I'll open up the browser again, and we can look at the different compounds that are being generated. So once more, I'll change the rendering and put this ball and stick in gold, and we'll rotate here just to have a quick view. And so you can see the first compound has an oxygen where we had the acceptor feature, and it is making a novel interaction here, and NH here where we had the donor feature in our Pharmacophore, and again, we're making a novel interaction at this position. So now I can browse through the structures and see if I find some interesting ideas, some compounds I want to work with. So that basically summarizes how you can run the add group to ligand application in Mo. So let me close this. We'll stop the search and we'll move on to the next example. So in this case, we only had one fragment bound to this receptor. But in some cases, what you might have, and I'll just go ahead and open up my system manager, is a second fragment bound to a different part of our pocket. So in this example, we actually have two fragments and we want to link them together. So how do we actually create a linker that goes from one to the other to create a single molecule? So that's what we're going to look at in this next example. Now, before we do that, as we're thinking about linkers or groups that can fit in here, perhaps we want to know a little bit more about the properties of our pocket. And so one way to do that is to go back to the Surfaces and Maps panel and to generate what we call an electrostatic map. And in fact, it's three maps. It's a hydrophobic map, an acceptor map, and a donor map. So I'll use all of these defaults here, and within 6 angstroms is fine. I'll click Create. And as you'll see, there's going to be 3 different maps: hydrophobic maps in white, acceptor maps in red, and donor maps in blue. These calculations are really done without any ligand present, and they're going to tell me what are the hotspots, the areas in the receptor where the receptor would like to see hydrophobic, acceptor, or donor groups. So let's have a look at each of them. I'll just make the hydrophobic maps green so they pop out on my white background. I'll change the rendering here, and we'll look at each of them one at a time. So let's start with the donor map. So this is, again, telling me where the receptor would like to see hydrogen bond donors in a ligand. So for example, if we zoom in and focus on this side here, if we were thinking of optimizing this group here, perhaps we could add something here and try to go position a hydrogen bond donor here. So that would be one option. On the very other side here, where we saw that we could perhaps build something bigger where we have the phenyl ring, another thought might be to position a hydrogen bond donor to create a new interaction with this blue patch right here. And of course, if I rotate, we can see that this corresponds to the position of this aspartate group right here. And again, if I rotate some more, of course, where we position our Pharmacophore feature for a hydrogen bond donor, there's also a blue patch right here. So those are the ideal locations to position hydrogen bond donors. I can look similarly at the acceptors. So of course, here we have one right here, which this fragment is already matching up with. And there's a red one right here as well. So this would be an ideal location for an acceptor, and we've already positioned our Pharmacophore feature right there. Now, the one I'm really interested in showing you right now is the hydrophobic maps, because we're interested, again, in building some linker to connect these two. And turns out if we look at the middle portion right here, we can see there's 2 green patches. So as we're building a linker, yes, we want a donor and an acceptor, but we probably want some hydrophobicity as well. And if we rotate, we can see there's actually 2 hydrophobic patches here, one on the top, one on the bottom. And so this is something we're going to target. So as we're looking through the different linkers that are going to be generated, we can have a look and see if we're finding some hydrophobic groups. Okay, so that being said, let's set up the calculation. So I'll minimize this. We'll go back to our panel, and we're going to switch now the operation from add group to ligand to link multiple fragments. So all I need to do is select the attachment point. So I'm going to select on this side this attachment point. And you'll notice the linker or exit vector is now red, not green. That just means my query is invalid, or in this case, incomplete, because of course I need to select attachment points on both sides. So I'll rotate, I'll select an attachment point on this side as well. And now the exit vectors are green, which means it's a valid query. Now, as I rotate, you can imagine I selected this hydrogen, but I could have also selected this hydrogen because it's also pointing in the right direction. So what I can do is I can actually select both of them. And as I do that, you'll notice the connection vectors are actually yellow now. And yellow means optional. So at this point, I have, if we look at the panel here, 2 fragments, 3 connectors, only one is essential, and that would be the green one here. In other words, I'll be looking for linkers, so fragments that can connect from here to here, or from here to here. And in fact, I could be connecting from here to both here and here and creating a new cyclic structure. Okay, so that's going to be our query. Now, as I rotate here, we can see that the tunnel, the space where we're going to fit our linker is quite tight. And so it is possible that a lot of the linkers are actually going to be clashing with the protein environment. But in reality, we can imagine that there might be some movement in the binding site that can accommodate a larger linker. And so to try and compensate for that, what we'll do is we'll just allow for a little bit more of protein ligand clashes, keeping in mind that we can then minimize our receptor structure as well. So to do that, I'll just go to the configure button here, and I'll just increase the protein ligand clash energy threshold. Now, that being said, we can also minimize the structures as they're being generated directly in the binding site. And so we'll do that by toggling on this minimize generated structures option. This is going to do an energy minimization of the ligand in a rigid receptor environment. And finally, since I'm minimizing these, I might as well score them as well. So I'm going to select GBVI WSA, which is the same score as we use in our docking application. So that being said, let me just change the output database name so we don't overwrite the last one. I'll set it to link.mdb and I'll go ahead and click search. So as I do that, the database viewer again opens up with my new currently empty output database. And so the hits are now going to be written out as they come in. And if I move to the side here, we can see there's going to be a new field called GBVI WSA. That's going to be our final score once we've minimized the ligands in the pocket. Okay, so as the hits are coming out, let's open up the browser once more to have a look at some of these hits. I'll open the browser. Now, as I scroll through these, and I'll just change the rendering once more, I'll make them gold again. You'll notice that a lot of the ligands are going to be linking out of this position, but some of them might also link out of this position. Remember, we had two optional exit vectors. In addition, you'll notice that the fragments are not completely on top of the initial structure. If you remember in the first example, I had pointed out that the fragment here was exactly in the same position. Now, that's not because we're doing the linking, that's simply because we've now turned on the energy minimization. And so once we've linked the two fragments together, we're doing an energy minimization. There might be a little bit of movement as the ligand adjusts to the protein environment. Okay, so let's have a quick look at some of these hits. So this is the first one, and I can use the arrows again to browse through different hits. And for now, all of them are linking from the top position, but as I go through, for example, this one here is linking through the bottom section. Now, I was also interested in finding linkers that would hit either of these two hydrophobic patches, and so I can scroll and see if I can find some interesting ones. For example, this one here is clearly hitting this hydrophobic spot right here. It's not hitting this one, but if I rotate here, it seems like a good structure because these two here, these two hydrogens are pointing right in this direction. So I can imagine maybe further optimizing the scaffold, positioning something here or here to actually fill this hydrophobic region as well. So that's the idea with linking. Of course, if I find a compound that I like in the database, they're all saved in the database, but if I want to keep it in my system manager, I can just go ahead and click keep here in the top right corner. And now when I close the database browser, you'll notice it's kept in the system manager. And so now I can continue working on this specific compound. Now, let me rename it because just so I can remember which one this was. So I'll call this link since we obtained it from the linking. Okay, so that being said, let me close this database and I'll stop the search. So the next tool I want to show you is scaffold replacement. So the idea here is, let's say you have a compound. It could be this compound, and I'll just hide the initial fragments. So this compound, for example, that I've created using the linking tool, but it can also be a co-crystallized ligand. It can be a ligand that I've docked to the receptor. Once I have this, how can I actually explore replacing a central core, a central scaffold in my compound for something else that might be have better binding affinity for my receptor, or better properties, and so on and so forth. So for that, we have a tool called scaffold replacement. Now you'll notice as I click here, there's actually two modes for scaffold replacement. The first one is select scaffold, and the second one is select R atoms. So I'll show you both of these really quickly. I'll start with select scaffold. So let me just hide perhaps the surfaces for now, just for clarity. We can come back to these a little bit later on. Okay, so the idea with select scaffold is I select the atoms I want to replace, and then I'm going to be looking for scaffolds that fit in the same geometry and that can replace the central core. I can do that in the main window here. What I'll do actually is I'll minimize this, and I'll show you how we can do that from the 2D depiction, which is available by clicking on the 2D button located near the bottom right corner. So the idea is you can draw a selection box around the atoms that you want to replace, so that central core. When I let go of my mouse, these atoms are now selected with the pink spheres. And in the main window here, you'll notice they're also selected, and we have this green halo, which is showing what's the scaffold I want to replace. You'll also notice the same green vectors on both ends here. Those are the attachment points or the exit vectors. And so we're going to replace the scaffold with something else, keeping the geometry intact at these two positions. So that's the first way we can select a scaffold. The second way, if I go back to the panel here, is by selecting our atoms. So the idea, and again, I'll make this smaller so we can view the 2D depiction. The idea with select our atoms is instead of selecting the entire scaffold, we're just going to select the endpoints. So for example, I can say my scaffold goes from here to here. And when I do that, again, the scaffold is highlighted in green with the green R vectors. Now, one of the advantages with this mode here is that I can actually connect in multiple different positions. And so, for example, if I'm not sure if I want to go from here to here or something smaller, I can select multiple endpoints like this. And so here I'd be doing the equivalent of 4 different queries at once, from here to here, and so on and so forth. And I could also select multiple endpoints here. And in the main window, you'll notice now I have different exit vectors that are highlighted. So that's the idea. So that's what we're going to do right now. So otherwise, I'm going to leave all the defaults here. The only thing perhaps I'll change is the molecular weight. I can see that my molecule right now has a molecular weight of 461 already. Since I'm trying to find potentially bigger scaffolds, I'm going to increase the size, maximum size to 700. I'll change the output database as well. And we'll go ahead and click search. So now the scaffolds that are being found, so those linkers that are going to be found, are going to be of different sizes because I do have different endpoints. But nevertheless, the idea is the same. It's going to replace a central core in my molecule. Now, for the purpose of this webinar, of course, I keep on clicking stop search because the purpose is not to run to completion, but typically you'll run this to completion and maybe you're running on a much larger database. And so what you would typically do, because you might have hundreds or thousands of different hits, is to sort by the GBI WSA score. So I'll show you how you would do that. I'll stop the search because we are running out of time. I'll go to the GBI WSA column. And so typically what I would do is right click, sort ascending. That way, the compounds with the best value are at the top here. And so now I can go ahead and browse these. And the best ones are going to be the first ones that I'm going to view. And so if I have thousands of compounds, I don't need to actually look at all of them. Okay, so let's have a look. I'll hide the initial structure here and maybe bring back my surfaces really quickly. There we go. So we can see we were aiming, if you remember, to go capture maybe the hydrophobicity at the bottom here, and we can see that clearly some of these better hitting compounds, the ones with the best GBI WSA score, actually do that. And so I can scroll, and again, if I find one that I like, for example, this one is matching up both of these hydrophobic regions, and in addition, it's making an additional hydrogen bond here. So if I like it, once more, I can click keep, and then I have that compound to work with in my system manager. Okay, so before we finish, I do want to show you just one last example. So let me just rename this so I can remember which one this is. And I'll just minimize it in the pocket, right? If I want to use this compound now as a starting point to do further modeling, it's always a good idea to hit minimize once more, just to minimize it within the receptor, right? So far, when I was minimizing the structure, it was rigid receptor environment. Now you'll notice slight movements of the side chains because initially when we prepared the structure with the quick prep application, we set some tethers so it doesn't move too much, but they're not completely fixed. Now that I have this optimized structure, the last example I want to show you is how I can use MedChem transformations to further optimize this phenyl ring right here. So if you remember, we saw that there was some extra space here to build off. We also saw that there was some great big donor patches here. And so there's definitely some opportunity to optimize this section of my ligand. So for that, let me just hide these. We'll go to our panel and we're going to switch to MedChem transformations. Now, like I said, I'm just going to focus on the phenyl ring. So I'll go ahead and select the phenyl ring and we'll run the application. I'll just change the name here to MedChem and we'll run it. And while it's running, I'll tell you a little bit more about what's going on. So far for the different modes that we tried, so the add group to ligand, the link multiple fragments that replace scaffold, we were always using the same linker database. Now, in this case, we're using MedChem transformations, and so we're using a transformation database instead. What did this look like? Well, I'll just open it up to show you. I open the browser here. These are sets of transformations, so transformation rules based on medicinal chemistry or intuition, which encode changes that we want to make. So basically, for any of these molecules, for the molecule I have here, anytime it finds one of these fragments, it's going to generate an analog of the molecule, which has just this minor modification in it. And you can customize this. So we provide this list, which has 175 different transformation rules, but you can provide your own. You can add them to this list, or you can create your own database of different transformations, for example, based on bioisosteric replacements you like. And then the idea is that you're going to get an output database which has just these minor modifications. So let's stop this and I'll show you really quickly what that looks like. So if I open up the browser and I'll change the rendering once more. Put the screen. If you remember, we had the phenyl ring that we were modifying. And so now, for example, we're going to see different positions of that nitrogen around the ring where we had just a phenyl ring. And then we're going to see some fused rings like this, and then perhaps some 5-member rings as well. And so it's going to make just those minor modifications. And of course, when I find one that I want, I can click keep and further minimize it in the pocket to see if I can achieve novel interactions of interest. And so, for example, here, as I minimize it, we can see we're capturing that new hydrogen bond we were aiming for. So that being said, I think that's we're almost out of time. So I'm just going to finish with a few slides just to recap, summarize what we've learned today. So we looked at using add group to ligand to grow a fragment. So how can we select an attachment point on a ligand, on a bound fragment, and actually build into a secondary part of our pocket? We looked at link multiple fragment, which is a way to connect 2 different fragments or more than 2 different fragments in a binding site to generate one single molecule. We also looked at using Pharmacophore queries to target specific interactions as we're building novel compounds. We looked at scaffold replacement, where the idea here is to select a central core of our molecule and generate analogs where we're going to replace this scaffold, the central core, with something else. And we looked at 2 modes for doing that, selecting the entire scaffold and selecting the R group position. And finally, we looked at medicinal chemistry transformations, which is based on rule-based medicinal chemistry rules or transformations or reactions, where we're going to do very minor localized modifications to generate a series of analogs to try and optimize, for example, the affinity or the properties of our compounds. And throughout, of course, we can use 2D filters, 3D filters as well. And so in this last example, we tried to optimize this phenyl ring using medicinal chemistry transformations. So that being said, I think that's the time we have. So Barbara, I don't know if we have time for a few questions.
Thank you very much. Well, I think we should really skip the live questions today because we're really running out of time, but be ensured that we have made notes of all the questions and you will receive answers by email. If you have any additional questions or ideas maybe for future webinar topics, you can also send us an email to support@chemcom.com. You see the email address on the last slide here. And I'd also like to direct your attention to the list of upcoming webinars and virtual workshops. So there's much more to learn in the next few weeks. And the next one is actually tomorrow. There's a virtual workshop on applications of pharmacophores and drug design. Thank you again, Val. That was great. Thank you for joining and listening, and goodbye.